Hi, remote students. I'm going to be going over Chapter 10, Lesson 1, Discovering Cells. I'm going to be highlighting and going over the answers in hopes that you could double check yours. Remember, it's really important that you do your questions first. I'm sure you could wait until I answer them, but it's not helping you if you do that. You kind of want to check to see if you found out the same important material or that how you process the information, I guess. But let's get started. Chapter 10, Lesson 1 is on discovering cells. There are three main points that they want. First of all, just what are cells? What is the cell theory? And how do microscopes work? You probably have already watched this video, so you have some idea of what's going on here. A little bit of My Planet Diary. We thought that was kind of cool. Why was he so excited about what he saw? The kids thought it was because it was brand new to him. I'm going to just fill that in. Somebody asked me to use pen here. It was brand new to him. Imagine if you opened up your closet and then you found a little village of little creatures that you didn't know lived in there. It would be kind of exciting. So remember, we want to highlight all of those bold words. Cells form the parts of an organism and carry out all its function. So you can see that form and function. So over here, they illustrate that. Here's one cell from this skateboarder and the actual skateboarder. In both the entire organism, this kid and this cell, they take in food, they let out waste, they take in water, they let out any excess water they have. They bring in oxygen and they get rid of carbon dioxide. So the same thing over here. When we got rid of carbon dioxide, we spoke a little bit. Let me go over this about how to write some of the chemical things, because chemistry will be our last subject, I guess, that we're going on, because believe it or not, the end of school is already right around the corner. So I don't know if you can see that, but these two are kind of coordinate. So if you struggle seeing my answers, the right answer is there because these are the same. So this is excess. And I did H2O, sorry about that. So down here, an organism's functions are the process that just allow it to live, grow, and reproduce. And these include obtaining the oxygen, obtaining food, and getting rid of wastes. So what this says right here is what the picture is illustrating to you. So the first part of lesson 1 says, now I get it. I know that a cell is the basic unit of structure and function. You would not have any bones without bone cells. You wouldn't have any muscles without muscle cells, and they carry on all the functions. So turn the page here. This is, I thought that video that we said of the cartoon, the kids really enjoyed it in class, but what is the cell theory? Let me keep this on the camera here. Just kind of what a microscope is. It's a tool. Most of the kids knew that. I wanted to point out that this guy, Anton van Leeuwenhoek, was the first to see living cells through the microscopes. But let's go a little bit through the cell theory. This man right here, Robert Hooke, isn't really credited or remembered, but he was the first person that saw a cell, and he also named it, but he used cork. So his were dead. He didn't see any movement. And then this guy was the first to see living cells. So we wrote down kind of like that they moved, that living things moved, that they were, living things were there, I guess, that were moved and were small. He was the first one to see living ones. In fact, we didn't actually know they were cells, that he called them animalcules, meaning very little animals. And then these three guys came along, and if you watch the video, you thought it was funny because these two liked each other, and then they argued with each other, and then this guy came in and cleaned up the argument with them, but he used another scientist's information, so then they started arguing. And if you are familiar with the man named Sir Isaac Newton, like in the video, didn't really get along with this guy because they were partners initially, and he felt that Sir Isaac Newton had taken his information, or he did part of the work and Newton didn't give him any credit. So then when they were both sort of head of the Royal Society in England, when Newton came after him, Newton sort of erased him. And if you're not written down, I guess people don't remember you. So here's the three basic parts of the cell theory. If I give you a question on this, if I give you a quiz, this question would probably be on there. Just living things are made of cells. Cells are the basic unit, and all cells are produced from other cells. We kind of like this because the guy that realized animals had cells was named Swan, and we think of Swan. And then I think of all of these as big trees in a forest. So this guy was a botanist, and he said, hey, I think plants are made of cells. And this is the guy that came up that said that you need a cell to get a cell. So let's check these. Oh, over here, fill in the circle. Who was the first guy to see living cells? So it would be here, not Robert Hooke. Why would Hooke's discovery have been impossible with a microscope? Because a human eye cannot see objects that small. Okay, use his ideas to explain why plastic plants and stuffed animals are not alive. Plastic objects are not made of cells. Thank goodness, because my daughter's stuffed animals would have reproduced, and we kind of would have had a hoarding situation then. And then the last one here, now I know that the cell theory describes, says, I don't know that how cells relate to, and we just came up with the book because I like to answer without using the teacher's book. The teacher's book says how all cells related to the structure and function of living things, but we just wrote how cells relate to living things. But I'm just going to write the living then with everything we saw. Okay, so how do microscopes work? Unfortunately, when you're at home, unless you have your microscope, I'm not telling you to go out and buy one or order one. So I have a video on there. In 7th and 8th grade, I think you also get an opportunity to use them. But so we can highlight this key idea here that some microscope focus light through lenses to produce a magnified images, and other microscopes use beams of electrons. Now, the electron microscopes are pretty much relegated to very fancy colleges and hospitals and things like that. They're very expensive. But the book gave us 3 magnified objects, and they just asked you to think what they were. This one, somebody said that it looked like a feather. There was another one, a lot of people that thought it looked like certain materials, and what made you think of that, and we were just talking about the pattern. I got an orange, and once again, the dimples look familiar. We also got a close-up of sauce cooking on the stove. This one, everybody seemed to think was a butterfly wing. And once again, the color and pattern were familiar. Okay, so we did this part down here. We didn't do this part up here, which is letting you know that the prefix magni or magna means big or large, making things larger. And it asked us to go through here and look at, underline all of the words that means, that had the word magnifying in them. And here's another one that means how you can enlarge it. So over here, magnification with a compound microscope, which is what we're going to be using. I drew the attention that you guys know what a compound word is. It's a word that is made of two words. A compound microscope has one and usually two lenses. These are different powers, but you use two lenses at one time. You might be able to think of something along the lines of having something like this and then having something like this and actually getting it to focus, which isn't going to be easy for me, and then the camera and that kind of stuff, which is kind of, that's what a compound microscope is. There's a way that you can figure out how much it magnifies by just multiplying. That's all it asks you to do here, multiplying to the lens. So if this increases things by 10 times, and then most microscopes have a 4, a 10, and a 40. So if you're using the 10, the one you're looking through your eye with, that's good. And if you have just the 4, and then you use the 10, and then you use the 40, how much would it increase? It would increase it 40 times. It would increase it 100 times. It would actually make it 400 times larger. What would happen if the object on the slide were too thick? Light can't shine through. That's an important part of using our microscopes will plug in, and this is a light. Some of the simpler microscopes have a mirror down here that you just want to reflect the light up there. Light can't shine through. Basically, what you see is you just see black. Now, this is really cool, and this is, I don't want to tell you this, I don't know if you want to read it, this is a dust mite, and they live in, just so you know, everyone's home. This is a colorized image, and it's made with an electron microscope, which you can kind of see all these different details. I always think that when people are making monsters in movies, all they are is really using microscopic images of maybe things that already exist. But over here, when you're looking at a microscope, this is a little bit higher, and I'm going to give you the answers here, but I just want to make it clear that I'm never going to ask you to do this. So when you measure things with microscopic, there are two things like magnifying. I think I highlighted that right here. Oh no, over here, I forgot to highlight that. For a microscope to be useful, it has to not only magnify, but there has to be resolution there. You have to be able to see the detail, because what would be the point if you just saw a big blurry thing? So these two things, so they're going to talk about these over here, and it's specifically resolution. So resolution is just the degree to which two separate structures that are close together can be distinguished is called resolution. Put simply, I think it's more like when you focus something so that you can really see the detail. So over here, when you're looking at a microscope or using a lens, and you're going to measure over here, the markings of the metric ruler in here, this is approximately 4.5 millimeters, 4.4. Look, you got 1, 2, 3, 4, and then that little bit more there. So I wonder if you can use this to estimate, if we were to actually use this and put that letter on there, how big really is this E? So I'm going to try to give you an example maybe that we could see here. I don't know if you could see a letter here. So here's that word. Let's say it's, what am I looking at? I don't know, maybe this word lab we can see. So lab, and we're going to make the L in lab. See, it's funny because I can't get you to see it as well as, so if you're going like, and see on the thing. So I'm going to make, so the word lab kind of fills up my lens like here. So how big is the real lab word? I know right now we can just move the lens and measure it at the metric side, obviously. But if it's teeny tiny like this, how do you figure this out? And just to estimate, I kind of slid this E over, I kind of went, okay, this E is the eraser and the metal thing here, and I brought the eraser and the metal thing over here, so it's going to be 1, 2. It's about 2 millimeters. So using a metric ruler, and can you figure out how much bigger is this E than the regular E? So what I did to just prove to you is if I go like this shows you that the E is about 2 centimeters. So if you know anything about the metric system, you need 10 millimeters to equal a centimeter. So this would be approximately 10 times, because this E is 2 centimeters, 2 millimeters to 2 centimeters would be times 10. And then over here, an electron microscope, how does it work? And electron microscopes use a beam of electrons instead of light to produce a magnified image. So they can actually, electrons are actually smaller than atoms. So let's go down and answer these final few questions here. The images in this color photograph actually consist of a few ink dots, and we kind of use this a microscope or a hand lens. I don't know, you probably won't be able to see that. We could actually see the dots here, but what color dots does an improved resolution have you see? And I think I can see red, I can see black dots, I can actually see blue or blue-green dots. That's basically how all colors are produced. When you look in here, you don't really see any yellow in here. And to me, if I was looking at this, I would have said yellow. Okay, so this just asks you to list 3 details that you can see in the photo. I would just think it like it has many, I don't know what those arms, you want to call them many arms. Okay, these look like they have, it has folds in the skin, and maybe it has feelers. I don't know what you would want to call those, maybe hair-like on the end of the arms. So getting these answers here, magnification makes things look larger. The diameter of a microscope's field of view is estimated to be about 9/10. About how wide is the object that fills 2/3 of a field? Circle your answer. I'm going to take some of these numbers down here. 2/3 and the number 9. I'm going to think of the number 9, 1, 2, 3, 4, 5, 6, 7, 8, 9, and take 2/3 of them, and that comes out to 6 dots. So I'm going to think that's my right answer, and it is. How are magnification and resolution differ? Well, magnification means larger, and resolution means more detail, I guess. You could say it's more focused. How do the characteristics of an electron microscopes make them useful for studying cells? They are more powerful. And show smaller objects, as we can see here. What I would ask you to do is look up magnified objects on the internet, and there's some pretty interesting things there. Now I know that a light microscope works by using lenses to focus light, and I'm going to introduce a word here. I've been saying it to the kids in the class. Focus light, that they bend it a little, and that when you bend light, that's called refracting light, and that would be a term that you should have learned in 5th grade, but with COVID we didn't get to the end, so I kind of had the kids read about it, but I didn't make them memorize that or anything toward the end. To refract light and making objects look larger. I'm just going to go ahead and say things look bigger. Okay, so when we go Thursday and Friday, we're doing a lab with the microscopes. If you have one at home, play around with it. If you happen to have two magnifying glasses, play around with it. And if you don't, please don't text me or email me that you don't have a microscope so that you can't do the labs. I'm going to be attaching pictures of perhaps what they're looking at under the microscope. Okay, so have a nice magnified experience.